This video is about how the digestion of lipids takes place inside the human body by the gastrointestinal tract. So, the digestion of lipids is different from that of the digestion of carbohydrates and digestion of proteins because the lipids are different from carbohydrates and proteins. The structure of lipids, as the lipids are insoluble in water. So the structure of the lipids have both the components. It has the hydrophobic and hydrophilic regions. So because of the complex structure of the lipids, as they are insoluble in water, so their mechanism of digestion and absorption is different from that of the carbohydrates and lipids. So from where we get the lipids? As we get the lipid in the form of food. So as you take the food that contains the lipids or the sources of lipids, lipid is the major macronutrient that are present in the food. So when you take up the food that contains the lipids, then they go through the digestive system where the digestion takes place, and after they are converting from the complex molecules into simpler when they pass through the different digestive processes and different digestive enzymes act on them, then the final simple products are then absorbed into the blood circulation, and from blood then they are taken to the they are reach to the they are transported to the different targeted sites and tissues of the body where they are utilized for their functions and where they are utilized for different processes of the body. So lipids, its digestion and absorption is different from that of proteins and carbohydrates because of its nature, that it's insoluble in water. They are just like waxes or just like the steroids and cholesterol, that they are insoluble in water, and they are soluble in same consistency like they will soluble in lipids and different solvents. So how the when you take up the food, they like lipid, you have to get the different energy sources from the lipid, as lipid is the highest energy source, and lipids has different functions in the body, that it provides the insulation and protection to the different body parts. So for to get the functions or to get these um, useful useful functions and processes from the lipids, it must be digested, it must be completely digested and absorbed by the body. So how the this food that contains the lipids, the in the form of the major or macronutrient, they pass through the different parts of the digestive system, as the digestive system is made up of different organs. So the food that contains the lipids for the purpose of digestion, to get the useful substances or the nutrients from that food source, it will pass through the different digestive organs. The major digestive organs are the it starts from the mouth, then the food that process by the process of digestion then pass through the stomach, then pancreas, and then small intestine. So these are the organs through which the food will pass and will be processed to be converted into more simple molecules to be absorbed by the small intestine and to be used by the human body. So what are the enzymes that will act on these complex molecules to be converted into more simple molecules of the lipids? So these enzymes that will be convert the complex lipid molecules into simple molecules, they will be lipolytic enzymes because they will cause the lysis of the lipids. So the enzymes used for the digestion of lipids are known as lipolytic enzymes, or also known as lipases. So there are different lipases that will be secreted from different organs of the body. This is the lingual lipase that is secreted from the mouth, from the sublingual gland. That these are the salivary glands that secretes the saliva and mucus and the um, different enzymes that are required for the digestion. So the enzyme that is required for the digestion of lipids in the mouth, that is the lingual lipase, that is secreted from the salivary gland known as sublingual gland. The other enzyme is the gastric lipase that is secreted from the stomach, and from the pancreas, the enzyme is the pancreatic lipolytic enzymes. These are many under the pancreatic in the pancreatic juice. There are many other enzymes are secreted that are involved in the digestion of lipids. The other is the bile salt is also important. Bile salt is basically secreted from the liver, as in the liver there is the gallbladder. Gallbladder is secreted from the liver, then collected in the gallbladder, and from the bile duct to the gallbladder it will enter into the duodenum where it will perform its function. So bile salts is basically involved in the emulsification of the lipids. Then the intestinal lipase that is released by the intestinal cells, that secus entericus, the intestinal lining of the small intestine, from where it will release intestinal lipases, and these lipases will digest or will break the complex molecules of the lipids into simple molecules to be absorbed in by the small intestine and to be taken to the blood circulation. So the organs and then enzymes are that are involved in the digestion of the lipids. So what will happen to the lipids as the complex form of the lipids are the triglycerides. As the lipids we take in the food, there are different types of the lipids. There are triglycerides, and besides triglycerides there are phospholipids, cholesterol esters, but almost 90% of the lipids that we take in the food is in the form of triglycerides. So most of the lipids that are taken up by the humans through the food are the triglycerides. So that's why the triglycerides lipids are then broken down with the help of the lipolytic enzymes, or you can say the lipases will break by passing through the different organs and through the process of digestion. They will be converted into the more simple molecules, glycerol and fatty acids. As triglycerides, they are made up of one glycerol molecule attached with the three fatty acids. So the enzyme that will break these bonds, the bond that links the lipids or that are involved in the formation of the lipids that will link the fatty acids to the glycerol are known as ester bond, and the process is known as esterification. So these enzymes, lipolytic enzymes or the lipases, they will break the ester bond present or involved in the formation of the lipids. So they will set free the glycerol and fatty acids. So these fatty acids and glycerol are the simple molecules that are released by the complete digestion of the lipids. So, in which organ, from which organ, which enzyme is released that are involved in the digestion of the lipids? The like mouth, stomach, pancreas, and small intestine. From the digestion of lipids is very less in the mouth and the stomach, but the real digestion of the fats, or you can say lipids, that's basically start from the pancreas, like small intestine. The pancreatic juice also comes in the small intestine. Then here the real digestion or the complete digestion of the lipids takes place, because here is the mechanism that is involved with the digestion of the lipids, because the digestion of the lipids cannot takes place in the other organs completely, or very partially, very little takes place in the other organs, because the lipids are insoluble in water, so the enzymes cannot easily act on them before they have to pass through the process of the emulsification. First they are converted into very small droplets, that is known as emulsification, that we will discuss in the upcoming. So mouth, it contains the enzyme lingual lipase that will cause very little digestion of the lipids. Then stomach contains the gastric lipase, as the enzyme that is involved in the digestion of lipid are known as lipolytic enzymes, or you can say lipases. Then pancreatic lipolytic enzymes are secreted by the pancreatic juice. Bile salts are basically come from the gallbladder through the bile juices that will contain the bile salts. Bile salts have very important function in the digestion and absorption of the lipids. Then intestinal lipase that are also secreted from the small intestine and present in the small intestine for the digestion of the for the complete digestion of the lipids into smaller molecules. So first of all, let's see each organ in detail, that in the each organ secrete which enzyme and how the digestion is carried out. In the mouth, the enzyme is the lingual lipase, that, as I told you, that very little digestion will take place in the mouth and the stomach. It's almost 10% digestion of the lipids takes place in the mouth and stomach, that they will convert in the large, very large complex molecules or very large droplets into small molecules, so that they can be easily digested in the other organs. So in the mouth, lingual lipase will act on it. They will convert the triglyceride as the most common form of the lipid taken in the food are the triglycerides. So triglyceride will then convert it into diacylglycerol and fatty acids. So as the glycerol, it is composed of three fatty acids. So it is all, that's why it is known as triglyceride. Then the lingual lipase will convert them into diacylglycerol and one free fatty acid. But this digestion is very rare. Then in the stomach, gastric lipase enzyme will act on them and will convert the large complex molecules into small molecules, so that in the small intestine, in order to convert them the large complex molecules into small, so that their complete digestion, to help them for the and preparing them for the complete digestion to be taken place in the small intestine. So it will convert this triglyceride into monoglyceride and fatty acid, but still there is no complete digestion. Like it was to be completely hydrolyzed or broken down into very simple molecules. Then the most important is the pancreas. The pancreatic juice that is secreted by the pancreas will enter into the small intestine, and there they will be involved in the digestion and breakdown of lipids into smaller molecules. So, pancreas, as it contains the pancreatic lipolytic enzymes. So, before the pancreas, as the bile is secreted by the gallbladder through the bile duct, the bile salts will enter into the small intestine. So, bile salts has a very important function, that bile salts is involved in the fat emulsification. So, what is emulsification? As fats are or the lipids are insoluble molecules. The example is here, as when you put the water and the oil in the glass, they are immiscible in each other. So they are immiscible, so the oil will float on the surface of the water. Same is just like a lipids, that they are insoluble in water, so they are not miscible into the water. So how the digestion will take place and how they are made uh, to be soluble in the water, how they will become soluble in the water, so that they must be taken up by the human body and they will have to circulate in the blood circulation to reach the targeted sites, cells, and organs. So how it will be possible? So this is made possible by the process of emulsification. So when you stir them, then what will happen, through a force or through some stir, then they will become emulsified. So emulsification is basically the small droplets, that if you make these droplets, as you have seen, that if you pour some drops of oil on the water surfaces, they will form a round droplets around the surface of the water and will float on the surface of the water. So this is the other example, as there is the water and oil, how you will make them miscible, how the oil will become soluble or miscible in that water um, environment. There is an emulsifier, as this is the emulsifier. When you stir them vigorously, then what will happen, they will form the small droplets. Still they are not totally soluble in this water, just like if you dissolve the sugar or salt in this water, they will completely dissolve in it. But in case of this oil droplet, they will still not completely dissolve in it, but they will form the small droplets, round droplets in it, in able to be miscible in that environment. So in this digestion process, as the bile salts are act as these emulsifier, as act as this trier. So bile salts will help them to become miscible in the in that environment, in that water environment, and will make them to be miscible and to be made them for the digestion. So as the lipids have both the regions, it has the structure of the lipids, as they are also made of phospholipids. So what will happen when the bile salts function is that it has the both region, the lipids have both region, as I told you before, that it has the hydrophobic regions and hydrophilic regions. So the hydrophobic regions means that it hates water, it does not like water. Hydrophobic region will arrange themselves inside, and the hydrophilic, that likes the water or that can survive in the water, they will be towards the water, they will outside the water. So they, when they the emulsifier is used, they will arrange themselves, these oil droplets or the fats or the lipids, arrange themselves into small droplets in a way that the surfaces or the structures that does not that are immiscible in water, like they don't like water or hate water, they will be from the inside, and the other that are miscible in the water environment, or that they like the water environment, will arrange themselves towards the outside, like hydrophilic regions will be towards outside and hydrophobic regions will be towards the inside. So through these soluble droplets, how what is the function of the bile in case of these lipids droplets? As the lipid droplets, this is the fat, let's suppose, and this is the water environment present in the human body. Now how, and these are the enzymes present. Now the enzymes, as they are involved in the digestion, how this fat will be digested, if they are not even miscible in this environment, how the enzymes will act on them. So to make miscible and to make this enzyme to act on these fats, what will happen, that the bile salts have this the property or the structural consistency just like fat, that the fat has also the both regions, hydrophobic and hydrophilic, just like the bile also contains the hydrophobic and the hydrophilic regions. So it will arrange themselves on the fat, so that they will become, they will made them emulsified. So these are the emulsification. So emulsification will break them into small droplets, so that it will make them, these will become into small droplets. So the bile that also contains the both hydrophobic and the hydrophilic regions will arrange themselves around the fats and will make them the small droplets. So these are the emulsification droplets. Now the enzymes, it these bile salts actually provide the site, as the enzymes will not directly act on the lipids, but the bile, when the bile cells bind on them, now it provides the opportunity for the enzymes to bind on them. Now with the help of the bile salts, the enzymes will come bind to these droplets and will hydrolyze these complex lipid molecules into the simple molecules, um, and to make and to for the complete hydrolysis, for the complete digestion of these complex molecules. So now the pancreas, now the emulsification takes place. Now the enzymes will act on these emulsified droplets. So what are the enzymes that are secreted in the pancreatic juice and taken up to small intestine by the pancreas? The enzymes involved, there are different enzymes released by the pancreas, as the lipids are not only triglycerides, but most of the, more than 90% of the food contains the triglycerides. It also contains the phospholipids and also contains the cholesterol esters. So the different enzymes will be secreted by the pancreas that will cause the hydrolysis, and that will cause the breakdown of these molecules. So this is the cholesterol ester hydrolase. So cholesterol ester hydrolase, it will break the bond in the cholesterol ester, and will form the, and will make the cholesterol free. So the fatty acid will be released and cholesterol will be released free. Then the other enzyme is the pancreatic lipase. So pancreatic lipase, it will break the triglyceride into molecule of glycerol and 3 fatty acid. The other enzyme is the phospholipase. So phospholipase, it will cause the hydrolysis or will break the ester bond present in this phospholipid and will release the lysolecithin and fatty acids free. So these enzymes, as the lipase enzyme, intestinal lipase, or these lipolytic enzymes that are secreted by these digestive organs, will act on these emulsified droplets. But there is other enzyme, that is the colipase. So colipase enzyme, that is also released by the pancreas. This enzyme is also required. This is a coenzyme that the other enzyme lipases will not act if the colipase is not present. So before that, the colipase will help in the action, to perform the action of the other lipases. So colipase will bind, it will provide the site for the binding of the lipases. So when the colipase will bind, then it will aid the lipases to bind and on these emulsified products, and to cause the digestion of these complex lipids. So when the colipase will bind, the colipase will help in the binding of these other digestive enzymes for the lipid, like it will help the binding of the lipase to bind on them and to break the complex triglyceride molecule into simple fatty acids and glycerol and cholesterol molecules. So this is the overall, that there were the large lipid droplets, but the digestion could not take place for these large lipid droplets. They have to first convert it into small droplets. So there will be the emulsification through the help of the bile salts. So bile salts, as these are the um, mm, lipids are insoluble, so bile salts are also having the hydrophobic and the hydrophilic regions. So the hydrophobic region will bind on them, hydrophilic will be outside, and through that hydrophilic regions there will be the binding of the enzymes. So these small droplets also increases the surface area for the binding of the and for the action of the enzymes. So they it will increase the surface area so that more enzymes could bind on them and convert them into more smaller products. So these bile salts will bind the enzymes. Pancreatic lipases will bind on them and cause the digestion. But before that there is the enzyme colipases, that before the colipases will bind, and colipases will locate the location for the lipases. So after colipases, the lipases will bind and then complex and then convert these and will cause the hydrolysis of these lipid molecules into more simple or complete hydrolysis of the or complete digestion of the molecules into small molecules. So in the small intestine, the enzyme is the intestinal lipase that is secreted by the succus entericus, that will completely convert the triglyceride into glycerol and three fatty acids, that will completely hydrolyze them or break them, convert them into glycerol and fatty acids. So this is the complete, like it will be hydrolyzing. This is the glycerol structure and these are the fatty acids. They are different lipids or triglycerides. They are different fatty acids backbone that contains the different carbon atoms. So if the fatty acids having the carbon chain like these carbon numbers, if less than 12 or if they are from 10 to 12, so these shorter chain fatty acids, they can easily cross the intestinal lining, intestinal membrane. They can easily be absorbed through the intestinal wall. But the large fatty acids, they has to pass through, or they will be absorbed to the intestinal lining through a special mechanism, through the formation of micelles. So at the end of the complete digestion of the lipids, the end product of the lipid digestion will be, there will be free fatty acids that will be present in the intestine. Okay, now after all these process, the complete digested product will be the free fatty acids. There will be the glycerol, monoacyl glycerol, and cholesterol. So these will be the end product of lipid digestion that will be present in the small intestine, to be taken up by the small intestine for absorption and to be carried to the blood circulation, to be carried to the other parts of the body. But there is special mechanism for the absorption, as the lipid absorption is different from that of the lipid from proteins and carbohydrates. So special absorption mechanism will be used for to be taken up these simple molecules to the body.